All right, so let's talk about the difference between exotoxins and endotoxins. Exotoxins are excreted by the bacteria into the surrounding environment of your body. Exotoxins can directly damage host tissue for the purposes discussed already, so that the pathogen can extract nutrients from the host tissues by eating the host, to tunnel through your body for spread, those kinds of things. So toxins that directly damage tissue are called exotoxins. Endotoxins are merely elements in the gram-negative bacterial outer membrane that trigger an immune response. So we have LPS, which are lipopolysaccharides. So that's specifically what an endotoxin is. So there's a lot of different kinds of exotoxins, so many different kinds that all do different things to damage your body. Endotoxin is specifically the lipopolysaccharide of the gram-negative bacterial outer membrane. Okay, that's what the gram-negative outer membrane is made out of. So when gram-negative bacteria die, their outer membrane starts to degrade. That releases this endotoxin, the lipopolysaccharide, LPS, into the bloodstream. And that signals to our bodies that there is a bacterial infection that needs to be dealt with and triggers an immune response. So endotoxins are not true toxins. They trigger an immune response. Our immune response makes us uncomfortable, but our immune response is supposed to be triggered by them. The reason our immune systems respond to lipopolysaccharides is because so many pathogenic bacteria have lipopolysaccharides. There's a lot of gram-negative pathogens out there. And so we have evolved to have an automatic inflammation and fever response to lipopolysaccharides. Okay? So inflammation response to bacteria is often a response to either endo or exotoxins produced by the bacteria. And both make us uncomfortable. Both, fever and inflammation make us uncomfortable. It's still good most of the time. It's good to deal with the bacteria. So endotoxins are just there to be part of the outer membrane. Just because they make us uncomfortable doesn't mean it's an actual toxin. It's a misnomer. So understand that endotoxin is a misnomer. Endotoxins are just the lipopolysaccharide from the outer membrane of the bacteria, and they make us uncomfortable because we have evolved an immune response. We recognize that we are infected when we encounter them. Right, an example of a very potent exotoxin is the botulinum toxin produced by Clostridium botulinum, or botulism. So this is the toxin that causes the disease botulism, which is characterized by paralysis. So when adults acquire botulism, it is usually not because they are infected by the bacteria, because the botulism bacteria does not take residence in healthy adult guts. However, if we eat food that had the bacteria growing in it, so also remember that botulism bacteria are not toxic unless they have a certain prophage in their DNA. So there's actually strains of botulism that are not toxic. So if we eat food that had Clostridium botulinum growing in it, that food could have the toxin from the bacteria in it, and so an adult can easily be poisoned by the toxin that's in the food. Okay, toxoids. Toxoids are non-toxic mimics of an actual toxin, and they're mimics of exotoxins specifically. And the reason toxoids are man-made. So the reason we produce toxoids is to stimulate antibodies that can neutralize the exotoxin. So when there is a bacteria in our body producing some horrible toxin that's making us sick, our immune system can generate antibodies that bind to the toxin and neutralize the toxin. But for some bacteria, like Clostridium botulinum, the toxin is, or Clostridium tetani for that matter, the toxin is so potent that it can kill you before you have a chance to produce any antibodies to neutralize it. So to deal with that, what you can do is you can go get a vaccine. So for diphtheria and for tetanus, the vaccine is actually a toxoid. It's a mimic of the exotoxin that does not actually cause damage to your body, but can stimulate the correct antibodies so that if you ever get infected by diphtheria or real tetanus, you already have those antibodies in your system. You can neutralize the toxin before it can make you sick. Okay. Okay. So, the other thing I was going to say about this is rattlesnake venom. So, rattlesnake antivenom is something that we produce through horses by exposing the horses over and over again to the toxin and continually increasing doses until the horse develops resistance. And that resistance is because the horse has made antibodies. Now, the horse has not created, this was not done through toxoids, but this is related to antitoxins. So an antitoxin is an antibody produced by a horse or some other animal that can neutralize a toxin if you're exposed to it. Okay? So a toxoid is something you give someone so that someone can preemptively begin producing antibodies by themselves to neutralize a toxin. So if you get a tetanus vaccine, that's what you're getting, a toxoid, a non-toxic mimic of the toxin. But let's just say you get infected by tetanus and you never got vaccinated against it. So you don't have antibodies to neutralize it because you never got any toxoids in a vaccine. Now you're being poisoned by Clostridium tetani exotoxins. What do? The answer is you get an antitoxin. So an antitoxin is the antibodies that some other animal made. So we usually make them with horses. So for example, an antitoxin for rattlesnake venom, you expose the horse over and over and over again to the venom. The horse becomes more and more resistant because it produces more and more antibodies. You extract the antibodies from the horse blood. If somebody gets bitten by a rattlesnake, you give them the horse's antibodies. The horse's antibodies neutralize the toxin in the person's body. Okay. And you can do the same for tetanus or botulism. So if you've never gotten a tetanus vaccine, you become exposed to tetanus, you can go to a hospital that might have an antitoxin for you. The antitoxin is the antibodies taken from another resistant individual's blood and then injected into your body to neutralize that toxin. So there are some strange microbes that produce what we know of as superantigens. Superantigens are exotoxins that create this massive immune response, way overblown immune response, so that your own immune system basically kills you. So the benefit to the microbe for doing this is not understood. We don't know why some microbes do this. It might just be some like weird mutation that just happens to do this and doesn't benefit anybody. We don't know. But so what happens is a superantigen is an exotoxin that indiscriminately stimulates T cells. So for a typical infection, 0.001% of your T cells will be activated. That's normal. So for just a normal pathogen, normal disease, 0.001% of your T cells will be activated to fight that pathogen. The reason such a few number are activated is because you have different T cells devoted to different pathogens. Every pathogen you've ever encountered in your life has an army of T cells devoted to fighting it should you ever encounter it again. So they're very highly specialized to individual pathogens. So a very small percentage of them are going to be activated by any individual pathogen. However, a superantigen activates a huge number of them. So a superantigen will activate 25% of your T cells. That is a way overblown. That's orders of magnitude higher immune response than is normal. So what happens in response to a superantigen is you get too many T cells activated, excessive cytokine production. Cytokines are the communication signals of the immune system. When one immune cell encounters a pathogen, it releases cytokines. Those cytokines attract other immune cells and activate other immune cells to recruit them to the battle. If you have excessive cytokine production, it means you're sending out these, every immune cell is sending out these crazy alarming signals to every other immune cell, and you have a way overblown response. And the result is nausea, vomiting, fever, and maybe shock. Remember, shock is a life-threatening drop in blood pressure. So an example of a pathogen that has superantigens is staphylococcal toxic shock syndrome. So women who forget to take their tampons out, they're at risk of staphylococcal toxic shock syndrome. So if you get too many of this genus of bacteria growing down there, this particular bacteria produces superantigens, and that can create this massively overblown immune response that's damaging to the host. So this is your own immune response killing you. Another example of a pathogen that did this was Spanish flu. Spanish flu caused a cytokine storm, a way overblown immune response. And it's the overblown immune response, not the virus itself, that killed the hosts, that killed the human patient. All right, endotoxins again. So remember, lipopolysaccharides are a major component of the outer membrane of gram-negative bacteria. When the immune system or an antibiotic causes death and destruction of gram-negative bacteria, the lipopolysaccharide pieces are released into the bloodstream, which causes a cytokine response, resulting in fever, diarrhea, general aches and pains, weakness, and in extreme cases, something called septic shock. Okay? So that's only in extreme cases. In normal cases, you will be uncomfortable from your immune response to gram-negative bacteria, and that is as it should be. If you feel uncomfortable, it means your immune system's doing its job. But if for some reason you're having an overblown response, you can have septic shock. So one of the ways you can get septic, so lipopolysaccharides are not a super antigen. The only reason they would cause an overblown immune response is if the infection was really advanced. You just have so many bacteria in your body. They are releasing so many lipopolysaccharides, and it's, because this is a graded response. When you just have a small infection, you have a small number of lipopolysaccharides, you have a small fever. You have a lot of gram-negative bacteria, you have a lot of lipopolysaccharides, you have a higher fever. So you have this graded response to the degree of infection. But if your infection has gotten super advanced, you've gone septic, you have bacteria replicating in your bloodstream, there's so many lipopolysaccharides being released all the time, your fever is getting way too high for your own body to endure. Okay? So if you give someone antibiotics for a gram-negative bacterial infection, you often see an immediate worsening of their condition as their body responds to the degraded pieces of the bacterial outer membranes. So at first, the person was feeling sick because you just have, the normal life cycle of bacteria. They live and die, live and die, live and die. So you have a smaller fever. If you put in this wave of antibiotics and there's this wave of death through the gram-negative bacteria, there's this wave of lipopolysaccharides in the bloodstream, you see an immediate worsening of symptoms in response to those lipopolysaccharides. So it's dangerous, actually, to give someone, if someone is septic with gram-negative bacteria, the gram-negative bacteria are all throughout their body, you give them an antibiotic and all of those bacteria die at once, that can be, the host's own immune response can be dangerous. So you have to be cautious providing antibiotics to someone who have a really advanced infection of gram-negative bacteria. All right, so response to high endotoxin concentrations. So again, this is, you have a huge number of gram-negative bacteria and they're all dying. So you have a lot of lipopolysaccharide, a lot of endotoxin. You can get endotoxic shock, which can be lethal. So remember, shock is a life-threatening reduction in blood pressure. Hypotension, that's just low blood pressure. Clotting factors might be activated, which can cause clots to form in your body. And that can be dangerous because it can stop blood flow through your veins and arteries. It can also, and those clots can also cause multi-organ system failure because those clots get stuck. Your organs are filled with tiny capillaries and blood clots have trouble moving through them, and so that can cause organ failure. All right, so the differences that arise in the different kinds of responses to endotoxins has to do with the severity of the infection, which is related to how many bacteria are in the body. I just really want to emphasize that. Endotoxins are not true toxins. They're only dangerous to us when there's so many of them because the bacterial count is so high in the body that our immune systems don't even know what to do with it anymore. Our immune systems just become overwhelmed. So this chart, it's not from your textbook, it's from a different textbook, but I like it because it really, it compares and contrasts exo and endotoxins, because I find no matter how much I say it during the lecture, students confuse it on the exam. So exotoxins, excreted by the bacteria for the purpose of causing tissue damage. Endotoxin, not a true toxin. It's part of the gram-negative bacteria outer membrane. More specifically, it's the lipopolysaccharide from the gram-negative bacteria outer membrane. All right, so that brings us to the end of chapter 13. We've covered portals of entry, or how a pathogen enters the body. We've covered adhesion, how the bacteria or pathogen adheres to host cells. We've covered how various microbes penetrate or evade host defenses, meaning the host's immune response. We've covered how and why pathogens cause damage to host cells, and we've covered how they exit.